Chronic diseases such as kidney, heart or circulatory diseases evolve often slowly and unnoticed. The treatment of these illnesses has been at the heart of 2 UN declarations. EU states have also joined this commitment, binding it under international law. Now scientists are applying a new method called proteome analysis to detect these chronic diseases in time and treat them successfully, and all they need is a urine sample. Urine contains proteins and peptides. They give hints how healthy our body is and whether an illness develops unnoticed. The method was invented by Harold Mishak.
So if we analyze the proteins, we can detect these changes at a very early point in time, even prior to any clinical symptoms. You can imagine that pretty much every disease starts, let's say, small, somewhere, starts with changes in proteins. If you can, let's say, cure this small lesion, then of course we can theoretically prevent every disease.
In our body, there are thousands of different peptides and proteins which regulate all biological processes. Changes in these molecules are often indicators of diseases. Detecting these variations will enable a much better diagnosis and early therapy. A single urine sample holds all this information. This new analyzing tool can identify more than 5,000 peptides and proteins simultaneously in one single probe and detect changes in their concentration. This is possible without any need for specific reagents, such as antibodies.
We have applied it by now for maybe 25, 30 diseases, something along these lines. But there is no logical reason why a specific disease should be excluded from this general rule that we first have changes in proteins, which then ultimately lead to the disease.
Over the last 16 years, Mosaiques Diagnostics has been collecting validated clinical history and urinary proteome data from more than 50,000 individuals. These data are now a key asset to enable distinguishing between similar diseases, even affecting the same organ. Today, the results of a proteome analysis can be obtained within only 90 minutes. The method is already used in Germany at the Hanover Medical School.
The patient quite often has additional problems with infections, with acute toxicity of the drugs, that early detection of the so-called graft-versus-host disease can be really difficult. And in this situation, the proteome analysis is really great because it's the best method we have to allow us to predict actually the occurrence of acute GVHD.
Proteome analysis can detect deadly diseases at an early stage and this way save human lives. The European Commission has already supported our research on several occasions and thereby recognized that this new method will soon change clinical diagnosis as we know it.